Good day, everyone. We are the group to discuss with you the Endoderm. Unlike most of you, we will be utilizing this media to supply our deliverables for ZOO 105. And like most of you, the pressure you get from Mom Edna's questions could translate to our patients, our frustrations in shooting apart multiple times, the noise of the street, the noise of the dogs and chicken, and the all-nighter editing. Nevertheless, the four of us developed this hybrid presentation hand in hand, the very reason why we have group reportings. For the objectives and outline, Jonald Pinongpong will discuss formation of endoderm and embryonic folding, yours truly on pharynx and GIT tract, Royin Radin on GIT accessory organs and respiratory tract, and Jay Postrado on congenital anomalies associated with endoderm. Let's get started.
So good morning everyone. My name is Jonald Kindo Pinumpong. I will be discussing with you the formation of endoderm, the embryonic folding, the anterior-posterior patterning, as well as the embryonic functions of the endoderm. So as we can see here, the formation of endoderm is not so different from the formation of mesoderm and ectoderm. It is the transformation of bilaminar disc to trilaminar disc in the process of gastrulation. Next scene. So as we can see here, the cells migrates from in the primitive strix or towards the primitive strix, forming the three germ layer. Okay. For the endoderm, it replaces the hypoblast and the following processes, which includes the formation of mesoderm, which fills the gaps between the hypoblast and epiblast. And finally, the remaining epiblast in the epiblast region will become the ectoderm. So that's how the endoderm form. How these cells migrate. So what is the mechanism that drives them to migrate? So this is due to the secretion of fibroblast growth factor 8 in the edge of primitive strix. They secretes this fibroblast growth factor 8 to activates the snail 1, which inhibits the eke adherence. So this eke adherence links these epiblast cells, and when this eke adherence are inhibited, they cannot link together the epiblast cells, thus creating the cells to migrate or making the cells to migrate towards the primitive strix, forming our three germ layer. Next. So as we can see here in the figure, from 14 to 15 days, the formation of bilaminar disc, it will transform into trilaminar disc at 16 days in the embryonic development. Next. So now we are in the embryonic folding, which is from flat trilaminar embryonic disc becomes a more cylinder or cylindric embryo due to the longitudinal and transverse folding that occurs as a result of embryonic growth, especially of the neural tube. So there are two foldings that will happen, they will form in this embryonic folding: the transverse or the lateral folding, or the longitudinal and the sagittal folding. Next. So for the transverse or lateral folding, there are two lateral body folds that will be formed. But we will not discuss this more kasi hindi naman mapapakita dito yung formation of primitive gut tube. So the best way to present the formation of primitive gut tube is the longitudinal or sagittal folding. So let's proceed to the longitudinal folding. So for the longitudinal folding or sagittal folding or craniocaudal folding is the folding of cranial and caudal, resulting in the formation of head and tail fold. So kaya meron tayong cranial fold and head fold. So this ito yung embryo na naka-fold cranially and caudally, incorporating yung yolk sac sa ilalim. Next. So in this section, longitudinal folding, when we cut that in median plane, so makita natin tong ectoderm, mesoderm, and endoderm, incorporating the yolk sac. So in the first embryonic folding, in the longitudinal folding, the endoderm moves towards the midline, still incorporating the yolk sac, creating the primitive gut tube that will further differentiates into foregut, midgut, and hindgut. For the foregut, it temporarily closed by the oropharyngeal membrane. It is located near the cranial end of the embryo, and the oropharyngeal membrane will eventually ruptures at the end of the fourth week of the embryonic development, resulting in the formation of the mouth. Next is the midgut, which is located between the foregut and the hindgut, and incorporates the yolk sac. Finally, the hindgut, which is located at the cloacal or caudal end of the embryo, which is temporarily closed by the cloacal membrane, which ruptures or eventually ruptures at the end of seventh week of the embryonic development. And this cloacal membrane will give rise to the urogenital openings and then the anal openings. We know how the primitive gut tube is formed from the bilaminar disc to trilaminar disc to trilaminar disc to and the embryonic folding happens and then the formation of primitive gut tube. Now we will be discussing the embryonic functions of the endoderm. So the first function of the endoderm is to induce the formation of several mesodermal organs. So these are the formation of notochord, the heart, the blood vessels, and even the mesodermal germ layers. The second endodermal function, the function of endoderm, is to construct the linings of two tubes within the vertebrate body. So we have the digestive tube and the respiratory tube. In digestive tube, it extends to the length of the body and buds from the digestive tube forms the liver, the gallbladder, and then the pancreas. From the digestive tube, it outgrows yung respiratory tube natin, it outgrows from the digestive tube and bifurcates or split to form the lungs. And anterior to the point where it bifurcates is mapoform yung pharynx natin. And finally, the third function of the embryonic endoderm is to form the epithelium of several glands. So the pharynx has this epithelial outpockets that give rise to the tonsils, to the thyroid, to the thymus, and the parathyroid glands. You might ask, how do some gut cells become pancreas cells while the neighboring gut cells become liver or intestine? This answers by the anterior-posterior patterning. So what is this anterior-posterior patterning? So this anterior-posterior patterning is the patterning, or it patterns the cells or group of cells into different kinds of organs along the anterior-posterior axis. So in this figure, the gut tube is divided into three regions by the gradient of WNTS, which is the wingless related integration site, the BMPS, the bone morphogenetic proteins, the FGFS, or the fibroblast growth factor, and the other paracrine factors, including the retinoic acid. So these are the paracrine factors that affects the formation of or differentiation of cells into different kinds of organs. So as you can see here, in the posterior end of the embryo, we have the more concentration of these paracrine factors. And highly exposed na mga cells to these paracrine factors will become the midgut and hindgut cells that will further give rise to the intestine. Above or in the middle of posterior region and anterior region, the cells there or the endodermal cells are somewhat lower na yung exposure nila sa ating mga paracrine factors. And these cells will become the posterior foregut cells that will give rise to the liver and pancreas. Finally, the anterior part or region of our embryo, which has the smallest exposure to these paracrine factors, will give rise to the anterior posterior cells that will further differentiates into the lungs and thyroid. The further discussion or further differentiation of different endodermal derivatives will be further introduced or discussed by the following reporters.
Good day, everyone. I am John Mark Polistico, and I will be discussing to you the endodermal derivatives, specifically the pharynx and the digestive tract. Okay, your endoderm forms the gut and respiratory tract in the trypoblastic organisms, just like as humans. So these are obviously essential for your gas exchange and food digestion. So your gut tube extends the length of your body and buds off from this tract gives rise to your gallbladder, your liver, and pancreas. These and the respiratory tract will be discussed by Royin a little later. But before we dive into the derivatives of the endoderm, let me introduce to you the very essential transcription factor that is essential in specifying the endoderm. This is what we call the SOX17. SOX17 in the study conducted by Hudson et al., they found out that this SOX17 solely controls the formation of endoderm in amphibians. Whereas in a Japan-based experiment, they found out that mice and zebrafish that has been deficient with SOX17 have endodermal or digestive tract issues as they progress to their development. This means that SOX17 is very critical information of endoderm. And as we progress with this report, we will also encounter different transcription factors and genes that are directing the endodermal cells in forming the primitive gut or your digestive gut. On week 3 to 4 of the embryonic development, the embryo or the trilaminar disc has established the body plan and it undergoes the transverse and lateral folding. This structure is the developing embryo in a sagittal section because sagittal section is very ideal in viewing your endoderm. If we take this green portion or that green lining and isolate it, we have this endoderm or your gut tube in cranial to caudal direction. Mesenchyme cells from the splanchnic mesoderm also wraps your developing endoderm. These endodermal cells will generate only the epithelial lining of your digestive tract. Meanwhile, the splanchnic mesoderm will develop the smooth muscle that will help your GI tract having its peristaltic movement. Here is our foregut, the midgut, and the hindgut. In this report, we will know what will happen to these three regions. Think of the food as you eat it. What is the first structure that it will go about? Of course, it's your pharynx. Next is, of course, esophagus. And just follow the path of the food. Here, the next one is, of course, your stomach. And this is where things gets interesting because not all structures are developed in just one of these three regions. Foregut actually forms only the first two parts of your duodenum, or how you pronounce it. That's entirely okay. You might ask, Mark, why is this happening? The division of the foregut, midgut, and hindgut is based on the arterial supply they get. The celiac artery provides blood for your what? Your foregut. The superior mesenteric artery, here, superior mesenteric artery, supplies blood for your midgut. And its inferior counterpart supplies blood for your hindgut. Mesenteric artery. Don't worry, guys. I will be providing the slides for this one with all these labels. We have the last two parts of the duodenum be formed in the midgut. Last two parts of duodenum. And just follow the path. What's next? It's the jejunum. And after that, we have ileum. So this portion is where your cecum, that's the next portion, and your appendix can be found. This structure, the upward motion, it's ascending. So obviously it's your ascending colon. Next, your midgut also develop or will give rise to the proximal 2/3 of your transverse colon. Only the proximal 2/3 of your transverse colon. Again, this is due to the arterial supply of these three divisions. Mark, why do these intestines or your small intestine, your colons have this convolution, or why are they intertwined? The answer lies on the midgut looping, in which your midgut rotates a total of 270 degrees. To know more about what I have just said, here's a video for you to visualize.
This is a brief overview of the embryological rotation of the midgut. Arterial blood to the gut tube is supplied by the abdominal aorta. The gut tube is composed of the foregut with blood supply from the celiac artery, the midgut with blood supply from the superior mesenteric artery, and the hindgut with blood supply from the inferior mesenteric artery. During the 5th week of development, the midgut starts to loop ventrally, herniating into the umbilical cord. This growth process creates much of the resulting length of the intestines. The cranial limb of this midgut extension will ultimately form the distal duodenum, jejunum, and part of the ileum. The caudal limb of this midgut extension will ultimately form the distal ileum, cecum, appendix, ascending colon, and 2/3 of the transverse colon. As the midgut lengthens, it also rotates 90 degrees counterclockwise around the axis of the combined superior mesenteric artery and umbilical cord. The cranial limb of the midgut, now on the right side of the umbilical cord, becomes highly convoluted throughout the next few weeks. The caudal limb of the midgut, now on the left side of the umbilical cord, grows a small cecal bud, ultimately becoming the cecum and appendix of the colon. During the 10th week of development, the midgut retracts back into the abdominal cavity and rotates an additional 180 degrees counterclockwise, for a total 270 degree rotation of the intestines. The duodenum, jejunum, and ileum are now inside the borders of the entire colon, which is composed of an ascending part, a transverse part, a descending part, and a sigmoid part.
Another question you might ask, Mark, why do the stomach, the pharynx, or the esophagus cells have different, or why are they different, or why are they unique from each other? The answer again is in the anterior posterior patterning, in which in the posterior level, high concentrations of this BMP, FGF, W or and WNT lies. And actually this WNT induces the transcription factor known as CDX1 and CDX2. At high concentrations, CDX transcription factors induces the formation of your what? Colon. And at a much lower concentration, these transcription factors induce small intestine. That's why they have different structure. It's because of the anterior patterning and the transcription factors that is acting on these cells. Okay, I think that's clear. Let's proceed now to the derivatives of your hindgut. You already have the proximal 2/3 of your transverse colon. So what's left? It's the distal 1/3 of your transverse colon. Next is your, here, it's the descending colon. Followed by what? Try to guess. It's sigmoid colon. Correct. And the last inches of your gut tube is the rectum and anal canal. Going back to our developing embryo, again, here we have a sagittal section. You can see here the cranial and the caudal end of our embryo. If we cut a section right here and somewhat like translate it into its ventral position, here we have these structures. We can see here a portion or a region in which an ectoderm and endoderm fuse. That is what we call the oropharyngeal pharyngeal membrane. That is one of the two regions or the two membrane where the ectoderm and endoderm meets. The other one is the cloacal membrane. Okay, focusing on the oropharyngeal membrane, as the development progresses, this portion or this primitive pharynx will have vesiculation here. That is the vesiculation that is forming, and in the internal portion or internal aspect, this primitive pharynx forms somewhat like pouches. Ayan. These pharyngeal pouches will give rise to the structures that we will be discussing later. So here's the first pharyngeal pouch, second pharyngeal pouch, third pharyngeal pouch, and your fourth pharyngeal pouch. Your first pharyngeal pouch will give rise to your, okay, I'll be needing chair for this one, your middle ear. Or your tympanic cavity. Next is the other one, auditory tube, or your eustachian tube. Okay, that's the first pouch derivative. The derivatives in the second pouch are your tonsils, the tubal, pharyngeal, what else? Yes, lingual, and your palatine tonsil. The third pouch will give rise to the superior parathyroid, inferior parathyroid, and your ear parafollicular cells, or your C cells. A bud off also will come off your pharynx. That is what you call, or that will become your thyroid. Another bud off will rise from your primitive pharynx, and that is actually your thymus. So if you have to add your derivatives, it would be thymus. The other membrane in which the ectoderm and endoderm fuse is the cloacal membrane here. This bifurcate posteriorly and anteriorly, giving rise to your urogenital sinus and anal canal. In the formation of anal canal, the ectoderm moves into the endoderm, forming this division or separation line called pectinate line, here in red. Superiorly, this is the 2/3 endoderm-derived anal canal, and inferiorly is the 1/3 ectoderm-derived anal canal. Your urogenital sinus will give rise to urinary bladder, urethra, and prostate gland in males.
Good morning, everyone. I am Royan Zinrisodin, and I will be discussing about the three accessory organs of the gastrointestinal tract. So the endoderm forms the tree lining of the accessory organs of the GIT. So the posterior foregut of our embryo contains progenitor cells that can give rise to pancreas, liver, and the gallbladder. So we can see at the foregut of our embryo, there were three diverticulums that will bud up from there: the respiratory bud or respiratory diverticulum, the hepatic bud, and the pancreatic bud. So based sa kanilang name, we can infer what their fate will be in the future. So let's talk about first the hepatic diverticulum. So the hepatic diverticulum have endoderm that will extend from the foregut out into the surrounding mesenchymes. And these surrounding mesenchyme cells will induce the endoderm of the hepatic bud to proliferate, to branch, and to develop the gallbladder. So liver formation. Heart cells promote while notochord prevents the formation of cells. So this condition is very important in the liver formation. So why is that? It is because of what we call the cardiogenic mesoderm. So this cardiogenic mesoderm contains FGF1 or fibroblast growth factor 1 and 2 to block the inhibitory factors that the mesenchyme induce on the embryo. So by blocking the inhibitory factors, it allows the proliferation of the liver-specific genes to be expressed in the region that it is intended to. But first, in order for it to react to the signals given by the heart, it needs to be competent enough to be expressed. So how can we make the liver-specific genes to be competent? So first, we have this thing called the forkhead transcription factors. So the forkhead transcription factors will serve as the leading transcription factors in order for the liver-specific genes to be expressed. So these factors involves the FOXA1 and FOXA2. So FOXA1 and FOXA2 will remove the chromatin surrounding the liver-specific genes in order for it to be more competent. And next, after the chromatin was removed, another type of forkhead transcription factors called the NH4A will serve as a driving force for the liver bud to be differentiated into liver cells. Next. So let's go back to this principle of anterior to posterior patterning. As we can see here, the hepatocytes or liver cells and the cholangiocytes that forms the cells of the bile ducts can be formed from hepatoblast. So the glucocorticoids, the FGH, FGF, OSM, WNT will induce the hepatoblast to be differentiated into liver cells, or what we call the hepatocytes. While the TNFB or the TGFA family, as well together with Notch factors, will cause the differentiation of hepatoblast into cholangiocytes or the bile duct cells. Let's talk about the pancreas formation. While the heart cells promote and the notochords prevent the formation of the liver, in the case of pancreas formation, it is the opposite. So the notochord actively promotes the formation of the pancreas while prevented by the presence of the heart. So why is that? It is because in the presence of the notochord in the region where the pancreas will form, the notochord represses the expression of the SHH or the sonic hedgehog protein. So it is a surprising finding since, di ba, we have talked about that the notochord is the source of sonic hedgehog. So that is true to the other case or other region of the embryo. But in the region where the pancreas will form, that region of notochord repress the formation of or the expression of the SHH. It is because in this region the notochord produces FGF2 or fibroblast growth factor 2 to repress or downregulate the expression of the sonic hedgehog. So next. So as you can see here in the illustration, we can see that at day 30, the ventral pancreatic bud is close to the liver primordium. So the most distinct feature that you can observe in the first 30 day of the illustration for the accessory organs is that the ventral pancreatic bud is separated to the dorsal pancreatic bud. So in this case, in the day 30, the ventral pancreatic bud is closer to the bud of the liver. Then the dorsal pancreatic bud is on the opposite side of the pancreatic bud. So at day 35, they start to migrate. It is because the rotation of the stomach occurs in this day. So as the stomach rotates, it brings the ventral part of ventral duct closer into the dorsal pancreatic duct. Then by the 6th week of development, the two pancreatic duct will start to be in contact, and at day 42, they will fuse together, forming the pancreas that we have now. So in humans, only one functioning duct will remain. That is the ventral pancreatic duct. And the dorsal pancreatic duct will just fuse together with the ventral, but on some populations remains two pancreatic duct to be formed, and that will be discussed by a deformities to be discussed by Janal Postrado. So next. The lack of SHH in the pancreas seems to allow in the region of the gut to receive signals from the blood vessels surrounding it. So as you can see in the illustration, we have three notable blood vessels that come in contact in the region and the pancreas forming region of the gut. So it is the aorta and the two vitelline veins. As you can see here in the staining of the histology image in the illustration, as you can see, the gut is in the middle and the top is stained blue. Aorta, then the lower two with stained blue are the vitelline veins. So these blood vessels induce PDX1 or the PDX1 gene in the gut epithelium. This gene is especially important in the formation of the gut. So some researchers tried to induce PDX1 into the liver forming bud, and they found out that this PDX1 causes this liver bud to form to de-differentiate back into gut epithelium, then differentiated again into pancreatic cells. So they have inferred that the PDX1 is the one responsible for the formation or the differentiation of the gut epithelium into the pancreatic cells. So next. PDX1. So generating of functional pancreatic B cells. In this study, they tried to convert the mature human skin cells into a functional beta cells. So the beta cells are the insulin secreting cells found in the pancreas. So first, they induce the adult skin cells with paracrine factors together with paracrine factor inhibitors in order to de-differentiate the adult skin cells into IPS, or the induced pluripotent stem cells. Then after the application of SOX17, the IPS then differentiated into definitive endoderm, then further paracrine factors induction, they become primitive foregut, pancreatic progenitor, then endocrine progenitor until it became the beta cells. And these beta cells was then injected into a diabetic mouse, and they found out an improve in the insulin levels of that mouse. So the gallbladder formation. So unlike the formation of the liver and the pancreas, very little is known about the formation or the development of the gallbladder. So actually, the most successful yet study conducted about the formation of the gallbladder is about its fate map that was discovered just this 2015 using the Dill labeling methods, or the staining of the Dill labeling. So Yumura and colleagues showed that the most of gallbladder progenitors were located in the lateralmost region of the foregut endoderm, and at the level corresponding to the junction of the first and second somites. So as you can see here in the illustration, we have the biliary atresia caused by the compound. So there was a crisis or a problem occurred in Australia. It is because during drought, they have observed that the livestock that they have in their farm have widespread carrying disease called the biliary atresia of the offspring of their livestock. So they studied and investigated, and they found out that because of drought, most of the plant fed to the livestock are dried out. So they chose to feed them with pig weeds. So this pig weeds contains the teratogenic compound called the biliaterosome, which causes then the biliary atresia, or when it is this disorder is called causes the lumen of the biliary to be blocked by the developing tissue. So let's talk about the respiratory tubes. So although they have no roles in digestions, the lungs and the respiratory tract are in fact derivatives of the stomach, or the GIT. So in the center of the pharyngeal floor, between the fourth pair of pharyngeal pouches, the laryngotracheal groove extends ventrally. So as you can see in the illustrations in the A, we can see the groove that forms at the fourth pouch of the pharynx. So this groove is called the respiratory diverticulum, or the laryngotracheal groove. So this groove then bifurcates into two branches, as you can see in the letter B and letter C. So these two branches will then give rise to the bronchi, the bronchioles, and the lung cells. So next. So let's talk about the problems involving the formation of that branches. So the tracheal esophageal fistula. So this type of disorder is the fusion of the two supposed to be different tubes of the esophagus and the trachea. So you can see in the illustration that the normal anatomy supposed to have two different tubes, and atresia, the tube of the esophagus is cut off, and the fistula, where the two tubes combined, and the atresia plus fistula, where esophagus was cut off, then it is connected into the trachea. So this type of disorder or deformities can be corrected using surgery methods. So the model for roles of WNT signaling and the BARX1 expressing mesenchyme in differentiating esophagus and the trachea. So we have observed that in the anatomy that there are two tubes entering our neck, so the esophagus and the trachea. So what are the signals involving the differentiation of these two tubes? So the absence of BARX1 protein, BARX1 absent, causes signals in the expression of causes the WNT to be present or to be expressed. Then the presence of WNT causes the expression of the NX2. Then this will cause the differentiation of the respiratory epithelium. While on the other hand, when the BARX1 is present, it blocks the expression of the WNT. Then it resulted in the transcription of the SOX2. Then the transcription of SOX2 will then cause the differentiation of the esophageal region. So BARX1 is absent, WNT present, cause the trachea formation. On the opposite side, BARX1 present, WNT absent, SOX2, then the esophagus will be formed. So let's go to the next part, the respiratory tube. So this image shows that it is the result when the right bronchial epithelium was allowed to grow and to be differentiated, while the left part of the bronchus was surrounded or induced by the tracheal mesenchyme. So they found that it seems that the differentiation of the tracheal and the lung cells depends on what type of mesenchyme they encounter. So, diba, as they bifurcate, they encounter different mesenchymes. So, they show here that when induced or surrounded with tracheal mesenchyme, the bronchus on the left can retain its original form and doesn't differentiate it further for bronchus into the formation of the lung cells. While on the right, where they allowed the bronchus to grow and develop, so we have here the expected results of the formation of the bronchus and the lung cells. So the induction of the lungs. Lungs are among of the mammalian organs to be last to be fully differentiated. The lungs must be able to draw in oxygen at the newborn's first breath. So to allow this to function, the alveolar cells produce or secretes what we call surfactants. So these surfactants cover the surface of the alveola in order it to prevent from sticking to one another and for it to collapse. So the surfactants actually, this is one of the surprising findings, the function of the surfactants and the development of the lungs. So it seems that the lungs are signals for the initiation of the labor during pregnancy. So they have found out that the surfactant A produced by alveolar cells in the embryonic lung tissue signals the dormant macrophages found in the amniotic fluid to be activated. So the activated macrophages will then migrate into the uterine muscles. The activated macrophages that migrates into the uterine muscles activated then the interleukin 1B or the I1B. Then this I1B produces the cyclooxygenase enzyme which then proceeds to the activation of the prostaglandins. So this prostaglandins will signal the uterine muscles to contract and will signal the start of labor for childbirth. So that would be my part in this report. Thank you.
I am Ginal Postrado. Today I will be discussing the congenital anomalies associated with endoderm. First, we have the pancreas divisum. Pancreas divisum is a congenital anomaly in which the ventral and the dorsal pancreatic duct are not joined together. As you can see here, the normal pancreas have two ducts, the ventral and the dorsal duct. As you can see, they are connected. Whereas here in the pancreas divisum, the two ducts are not connected, they are separated. As we all know, the pancreas secretes pancreatic enzymes which help in the digestion of food. An example of pancreatic enzyme is lipase, which works with the bile, which breaks down the fats. Another example is amylase, which breaks down the carbohydrates, and protease, which breaks down the proteins. 70% of the pancreatic enzymes are drained by the ventral ducts. It will pass through to the minor papilla and will enter the duodenum where the chemical digestion occurs. Whereas here in the pancreas divisum, majority of the pancreatic enzymes are drained by the dorsal duct. It will pass through the major papilla and we'll go to the duodenum. What is the cause of this anomaly? It is still unknown. So the symptoms, majority of the individuals born with pancreas divisum will have a mild or will not have a symptoms at all. In some cases, pancreas divisum is only detected during the autopsy. A small group of individuals will develop symptoms which commonly include abdominal pain, nausea, vomiting, acute and chronic pancreatitis. Next, we have annular pancreas. Annular pancreas is one of the most common congenital anomalies seen in our pancreas. The term annular pancreas means that there is a ring of extra pancreatic tissue that covers the duodenum. As you can see here in our figure, the pancreas surrounds the duodenum and it all goes up right here. So the pancreas can still function, but it can affect the function of the duodenum. For the newborns, they may not feed well. They may spit up more than normal. They not drink enough breast milk or formula, and they will cry. Up to one half of people with this condition do not have symptoms, or they will have a mild symptoms. There are also cases that are not detected because of the mild symptoms. According to Johns Hopkins Medicine, annular pancreas is associated with the Down syndrome because according to their data, 1/4 of the people with this condition have a Down syndrome. So next we have the choledochal cyst. Choledochal cysts are rare congenital dilations of the bile ducts. Dilations means enlargements. Bile duct is a network of long, tube-like structures that carry bile from the liver to small intestines for digestion, as you can see here in this figure. A study conducted in Japan revealed that 33 to 50% of the people with choledochal cyst are from the Japan, and majority of them are females. And the reason behind this is still unknown. So we have 5 types of choledochal cyst based on the site of the cyst or the dilation or the enlargement of the bile duct. So first, we have to familiarize the structure of the normal biliary tract. So as you can see here, this is the gallbladder, labeled as the number 1, and labeled in number 2 is our bile duct. So first, we have the type 1 choledochal cyst. It is the most common variety of choledochal cyst because 80% to 90% of the bile duct are dilated or enlarged, as you can see here in the figure. We have the type 2, isolated diverticulum protruding from the CBD. So the cyst protrude, as you can see here in the middle of the bile duct. It forms a balloon-like structure. So, para siyang balloon dito sa middle part ng bile duct. So next, we have the type 3 or choledococele. It arises from the dilatation of the duodenal portion of CBD or where pancreatic duct meets. This type of choledochal cyst is a severe type of choledochal cyst because it blocks the pathway of the bile duct papunta doon sa duodenum. So, ang tendency is yung bile is babalik doon sa liver and it may cause inflammation to the liver, pancreas, and the gallbladder. So, there is a high chance na magkaroon ka ng cancer sa liver, gallbladder, and pancreas. Next, we have the type 4 choledochal cyst. It is multiple dilations involving only the extrahepatic bile ducts. Extrahepatic means outside the bile ducts. So as you can see here in the figure, there are multiple dilations above the gallbladder. And there is also a dilation here at the middle of the common bile duct. So next we have the type 5 or Caroli's disease. Cystic dilation of intrahepatic biliary ducts. So ito naman is intra means inside. As you can see here, the cyst grows inside the gallbladder. And there are also multiple dilations above the gallbladder. So next. So what is the cause of choledochus cyst? It's a failure of recanalization of the CBD during the solid stage of development. As I mentioned earlier, pancreas has two ducts, the dorsal and the ventral duct. During the fusion at the early stage of development, the bile duct is at the solid stage. So recanalization is important because it is the process where the pathway develop. So doon sa pathway na yun dadaan yung mga bile and pancreatic enzyme. The recanalization starts at the middle of the our bile duct. Failure of recanalization may result to enlargement of the middle portion of the bile duct. So that's why the type 1 choledochus cyst is the most common kasi doon nag-enlarge yung bile duct. So what is the symptoms? Symptoms may appear in infancy or not for several years. Some symptoms include abdominal mass, pain in the right upper belly, jaundice, nausea, and vomiting and fever. Perforate anus or anal atresia. It is a congenital anorectal malformation wherein normal anal opening is absent at birth. As you can see here in the figure, this is the female and the male baby. Makikita ninyo na walang butas yung puwet nila or wala silang puwet. It is because of the abnormal formation of the colon. Fistula may be present in the vagina in girls or in the urinary tract in boys. Fistula means opening. So, there is an opening sa kanilang vagina for girls and sa penis naman sa boys. So, the tendency is doon dadaan yung dumi sa vagina nila or sa penis for boys. So, next. Unfortunately, the cause of anal atresia is still unknown, but it occurs in 1 out of every 5,000 infants. So the symptoms may have no anal opening, misplaced anal opening, no bowel movement within 24 to 48 hours after birth, and abdominal distension or bloating ng tiyan. With all this overwhelming information of our report, we will provide you the simplest summary of our report. Just remember the word endo. E stands for epithelial lining of gastrointestinal tract. N stands for the neck or the location of the thyroid, parathyroid, and thymus. D or the drainer, it represents the urinary bladder, urethra, prostate gland in males. And lastly, the O, which stands for organs associated with gastrointestinal tract, which includes the respiratory tract.
There must be another information overload going on your heads right now, and we might have memorized it ahead of you, but I know you can do it too. Thank you for listening.